basics and activity records in our laboratory. Just like we were starting from the first slides, and this slides contain four equipments. First of all, this one is autoclave that we normally use for sterilization of our media by biological waste and glass waste. We will use this autoclave. This one is the basic requirements. Second is the incubator. Incubators that we normally use for the incubations, for the cultivation of bacteria. Third one is weighing balance that is normally used for weighing of our samples, weighing of our powder media. We use this weighing balance and the biological safety cabinets. This cabinet is actually one of the most important requirements, especially when we deal with any kinds of bacteria. We need to make our environments clean. We need to work in the safer environments. And this biological safety cabinets that have the UV operations in its working that will provide a safer environments for our working. This one is the one of the highest needs of our microbiology lab. All of these equipments are also required. Now we go to the second slides in which we have different equipments just like magnetic stirrers, hot plates. This one is also the requirements which we use for the stirring of our media. We will do with the boiling of the media because most of the powder media is recommended to boil in the pH meter. That one is the most important for visualization of any media because if pH is adjustable within the range, the media showing its will show its characteristics. And the water bath for maintenance of media, you may place it for overnights on this water bath for maintenance of temperature that was recommended by most of ISO methods. Just like 44 to 47 degree, this temperature is recommended by the water bath. By the ISO method, then we will use this water bath and hot air oven. We normally use this one for our sterilization of glasswares and also for when we will wash this one, we need to open it so that any kinds of water residues will remove from this from our glasswares. And go to the next slides that contain all others necessary equipments for just like microscopes that you normally use for checking either cellular bacteria. You identified bacteria as gram negative or gram positive. Gram stain is also staining kits also requires for visualizations in the you can also check the motility of the bacteria one by an powder form media when otherwise is by using these microscopes. And others also an important equipment. This one is the colony counters that's also have a different upgradation now. Automatic colony counters also and manual colony counters. This one is just like a semi-automatic colony counters is required for counting of colony. We just need to place plates in the slide spaces given in the colony counter, and then we will check the results and we will place just add, we will subtract whatever we will visualize we need to count. We'll use this colony counters and the refrigerator. This one is most important also because most of the culture media supplements, plates or require are directed to store at this refrigerator temperature. Thus, we need refrigerators. Also for samples also. This one is also the requirements. Stomacher. This one is the requirements. Somewhere we use it called chooses a blender, but stomacher is also required for making homogeneous mixtures of our samples. After weighing the samples in our sterile bag, we simply match it in the stomacher so that uniform mixtures will be obtained for our testing. For further, we needed just micropipettes for transferring of cultures from one place to another and the thermohygrometer. This one is the most important thing. Also for a safer environments, we need to check our humidity levels, temperature level is according to the requirements or not. This one is all the equipment that we this one is all the basic equipment that we needed for our testing. Further, in the next video, we will go for the others basic other requirements just like glasswares and small other equipments that are required for our testing. That will also vary according to the parameters, parameter to parameters, but this one is the basic requirements for microbiology lab. Thank you so much.